A ribbon-cutting ceremony on the third floor of the Science, Engineering, and Technology building at Utah Tech University officially opened the new Center for Precision Medicine and Functional Genomics.
This center represents the peak of university and industry collaboration with our partners, Intermountain Healthcare, Intermountain Precision Genomics, and Culmination Bio. To find answers, we need bright minds trained in an area called functional genomics. At Utah Tech, we are training students in techniques that allow them to determine if a newly discovered mutation contributes to disease or if it is harmless to the patient.
To do this, students study and observe reactions in zebrafish.
The next question becomes, why zebrafish specifically? What's so unique about them? Well, it turns out that zebrafish, this little household pet that are often found in household aquariums, have a couple of unique features. First of all, they have genes that are very similar to ours. You can find, about 80% of the time, if you find a human gene, there is a homolog for that gene in the zebrafish.
Zebrafish, as I've learned more and more about zebrafish, I was teasing with Dr. Nadald when we were building this building. I said, well, what is it that you need? And he says, we need a zebrafish lounge. And he must have saw my face because I had no idea what that was. And I just confessed to him that I actually went and looked it up.
With using the zebrafish, we can identify the genetic, like a function, the gene function, or even like a mutation, which is a variant function in human beings.
On if these mutations cause cancer, cause various diseases, for us to be able to identify that and help the healthcare fields actually tackle those issues, I mean, it's immense.
This is the biology lab where functional genomics students study these freshwater fish. First, the scientists allow the adult zebrafish to mate. A female fish can lay hundreds to thousands of eggs at a time.
So males tend to be very slender bodied. They do have a bit more bright coloring. The females will have a large silver belly, especially if she hasn't propagated in a while. It'll be very, very large and pronounced.
Then they take the female fertilized eggs or embryos for study.
So right now what we're looking at is this is a glass tube that has been microheated and pulled to a point. We shear that down to a 0.2 nanometer opening. But this is the most effective way to inject embryos with anything, because embryos alone are very, very small.
Using a microscope, students inject the embryos of the zebrafish with a very tiny needle.
We thrust it into an embryo. We inject it with various materials, whether that contains mRNA or whether it contains some sort of CRISPR-Cas9 knockout.
To do this, they use what is called CRISPR gene editing, where they can knock out the bad gene. And because of the zebrafish's rapid development from embryo to fish, it's also faster to use in studies.
These are some baby zebrafish. We have a couple different sets. These guys are about 2 weeks old, and then these guys are about 1 week old, and you can actually see the size difference in between them two. You can also tell that they're translucent, which really helps with when we're looking, at these organs. So if we can see what organs are being affected from these variant genes quickly, we can have that data a lot quicker and send it to the hospital, to the other scientists, so they can go ahead and validate that.
And then we allow those to propagate, grow, and we start analyzing them physically, their physical traits, movement patterns.
We're able to say, hey, these proteins are different and they're not good proteins and it came from this gene. So if we can identify the gene that these proteins came from, then we can say we need to be on the lookout for this gene in humans, for example.
Those who stopped by the lab were impressed with the students' knowledge and equipment used in the studies.
This lab is amazing. I've never seen anything like it. And to stand there and try to look in the microscope and move the needle, it was hard, but I have no words. It's amazing. My mother passed away from ovarian cancer in her early 70s, and I wish that something like this could have been available to help her.
And others are supporting the students involved in this program with fellowship funding.
Dr. Nadald was my husband's oncologist, and So I became really interested in genetics and genomics and hereditagene and all of that when I met Dr. Nadal. My husband has since passed away from cancer, but it's been a passion of mine to further what he's doing here in the area.
I mean, as a student, there's the juggling between work and schoolwork. So being able to have this gift of being able to spend more time dedicated to the lab, it's irreplaceable and extremely flattering that you chose me. Yeah, thank you.
Oh, you're very welcome.
Major contributions for this new center came in part by community project funding from the federal government.
Representative Stewart and his office sponsored this program and made funding possible through the Department of Education. And Utah Tech recently received a $780,000 funding proposal in order to start this program.
And I want to quote the congressman. He said, "I am honored to have sponsored the funding of a new program at Utah Tech that will train variant scientists to play this vital role in the healthcare system."
So this partnership between Intermountain Health and Utah Tech and Culmination Bio is the culmination, essentially, of the effort to hand all of this information into a scientific setting where it can be investigated. And I think the best part is it gives great experience and opportunity for students.
In fact, the center supports a number of certificates. We have a certificate in functional genomics, one in bioinformatics, one in protein characterization, one in genetic sequencing, and we have a forthcoming master's degree level certificate in variant science.
It's amazing. The hands-on processes, what we learn and how to integrate what we learn and be able to apply it and actually make a difference. It's amazing.
So new that I just fell in love with it almost immediately. I knew I liked genetics a little bit, but the application of genetics is a lot different than just like the lecture of genetics too.
And the idea that we're going to take findings from the genome, understand them in little fish in this room over here, understand them in terms of the way we're doing clinical planning is exciting.
From Utah Tech University, Melissa Anderson, Community Education News.